Today's lecture will finish up Chapter 25 with Chapter 25, Section 4 with control of gene expression. Then we're going to go over and we're going to cover 25.5, which talks about gene mutations. Gene expression is responsible for making cells the types of cells that they are. Now, generally, all cells have what we call housekeeping genes. These are the genes that govern the functions that are common to lots of different types of cells. They're active in many cell types, and only certain genes are active in cells that perform specialized function. The gene expression is controlled in a cell, and this control accounts for its specialization. So if you can see down here in the picture, the gene type, the housekeeping gene, the hemoglobin gene, the insulin gene, or the myosin gene. Housekeeping genes are turned on in all three of these different types of cells. Those govern the typical things that cells need to be able to do. However, the gene for hemoglobin is what is prevalent in red blood cells, while the gene for myosin is turned on in the muscle cells, and the pancreatic cells or the insulin gene is turned on. Now, each one of those cells came from the cell that created your entire body and differentiated into these different types of cells. So they have the same DNA inside this nucleus as there is inside these nuclei and the nucleus that red blood cells ditch before they become mature, but that has the same set of DNA in it. So not all of those genes are expressed at the same time, only certain ones to make them specialized cells. And then you have the housekeeping genes that are on in order to have common things, proteins and things like that made that are characteristic of all cells. Prokaryotes are different than eukaryotes. Prokaryotes are bacteria. And before we get into how gene expression works in one particular bacterium, Listeria coli or E. coli, that you've heard of before that's in our gut, let's look at some basic definitions. First of all, an operon. An operon is a cluster of genes usually coding for proteins that are related to a particular metabolic pathway, along with a short DNA sequence that coordinately control their transcription. The sequence consists of a promoter, which is a sequence of DNA where the transcription begins, an operator, which is a sequence of DNA where the repressor protein binds, and those work together in order to give us gene expression. So let's take a closer look at this. So gene expression in the bacteria, the E. coli. Let's look at the lac operon. So what happens when lactose is absent? Now, lactose is milk sugar. So if you take in milk, you have a sugar that needs to be broken down. So when lactose is absent, what happens in the E. coli? The regulator gene codes for a repressor that's normally active. It doesn't need to metabolize lactose if lactose isn't present, so it's not going to allow that to happen, and it does that by a repressor protein binding to the operator. RNA polymerase cannot transcribe the three genes of the operon. The structural genes are not expressed, so they don't get expressed because lactose is absent. On the other hand, if you have lactose in your system, so you take in milk, here's what happens instead. The lactose binds with the lac repressor. The repressor is unable to bind to the operator, which is going to allow the structural genes to be transcribed and the enzymes be produced. The lac operon is considered an inducible operon, and the reason for this is because it's only activated when lactose induces its expression. So you have to have lactose around in order to cause it to be expressed. These repressible operons are usually active until a repressor actually turns them off. So let's take a look at this. This is what happens when lactose is absent. When lactose is absent, the active repressor is in the operator, and that causes the DNA polymerase not being able to bind and start the transcription of those genes. When lactose is present, it binds to the inactive repressor, causes it to release, allows the RNA to polymerase to bind to the promoter, and goes ahead and will transcribe the section of the three gene sequences needed for digestion of lactose. In bacteria, a single promoter serves several genes that make up what we call a transcription unit. In eukaryotes, it's a little bit different. Each gene has its own promoter. Bacteria rely mostly on transcriptional control. Eukaryotes employ a variety of mechanisms to regulate gene expression, so it's much more complex than in bacteria. It affects whether the gene is expressed, the speed which it is expressed, and how long it is expressed. Gene expression in eukaryotes. Let's see what happens as compared to the bacteria that we just looked at. Gene expression is controlled in eukaryotes by a few different methods of control, actually 5 that we're going to look at today. We can look at pre-transcriptional, transcriptional, post-transcriptional, translational, and post-translational control. In this diagram, you can identify where each one of these will take place. Pre-transcriptional, of course, is in the nucleus. We have transcriptional control, which is there again at the transcription level in the nucleus. Post-transcriptional control comes after transcription has taken place and the segment of messenger RNA has detached from the DNA. This can go through the nuclear pore into the cytoplasm and have a level of control called translational. And then we have post-translational control when we make a functional protein after this has been translated into a protein from the transfer RNAs working with the messenger RNAs in the process of translation. Take a look at pre-transcriptional control. This uses DNA methylation and chromatin packing to keep the genes turned off. Heterochromatin is an inactive set of genes that are located within a darkly stained portion of chromatin. An example of this is the X chromosome in females for mammals. In females, you know that we have 2 X's. Males have X and a Y. The males only have the 1 X. The females only have 1 X really that is active. And this inactivation of one of the X chromosomes happens in every cell inside the body. That inactivated X chromosome is called a bar body. These X chromosomes that become inactive, it's not always the same X chromosome inside each cell. So we see evidence of this in tortoiseshell cats. What we usually call them in our area are called the calico cats. So the calico cats that we see are females, and the reasons they show that pattern that's so odd is because different cells inside their body have different X's turned on and different X's turned off, and those that deal with the color of the fur, it's in a random pattern. You don't know which X is turned off and which is turned on. So if they're heterozygous for the color of their fur, which is an X-linked gene, the cat can be what we call a mosaic, or the tortoiseshell, or the calico. So they have patches of cells that are expressing different alleles, and this becomes very evident physically. So we can see it in the phenotype of the female cats that are that way. Let's also look at euchromatin. So the euchromatin are loosely packed areas of active genes, and they still need to be unpacked before they can be transcribed. And the chromatin remodeling complex pushes aside the nucleosomes. And once the nucleosomes are pushed aside, then the section of DNA that needs to be expressed can then be expressed, transcribed, and expressed. Just like in prokaryotes, in eukaryotes, transcriptional control is dependent upon the interaction of proteins with particular DNA sequences. And the transcription factors are going to bind to promoters to form the transcription initiation complex. The transcription activators speed transcription up dramatically by binding to the enhancer. Post-transcriptional control involves primary RNA process and the addition of a poly A tail and a guanine cap. We talked about during transcription how this has to happen in order for it to be ready to leave the nucleus and go into the cytoplasm for translation to occur. In addition to the poly A tail and the guanine cap, we have the removal of introns and the splicing of the exons together. The different patterns of splicing can occur during this portion of post-transcriptional control. Translational control finds differences in the poly A tails or the guanine caps and may determine how long a messenger RNA is available for translation in the cytoplasm. Specific hormones may also affect the longevity of the messenger RNA that's already been transcribed in the nucleus and is in the cytoplasm for translation. Post-translational control, some proteins must be activated after they're made, and many proteins function only for a very short period of time before they get degraded or destroyed by the cell. And that wraps up the control of gene expression for this chapter.